Welcome back to Concepts of Biology. We'll continue to read Chapter 2, Section 2.3, Biological Molecules. Lipids. Lipids include a diverse group of compounds that are united by a common feature. Lipids are hydrophobic, meaning water-faring, or insoluble in water because they are nonpolar molecules. This is because they are hydrocarbons that include only nonpolar carbon-to-carbon or carbon-to-hydrogen bonds. Lipids perform many different functions in a cell. Cells store energy for long-term use in the form of lipids called fats. Lipids also provide insulation from the environment for plants and animals. For example, they help keep aquatic birds and mammals dry because of their water-repelling nature. Lipids are also the building blocks of many hormones and are an important constituent of the plasma membrane. Lipids include fats, oils, waxes, phospholipids, and steroids. Figure 2.17. Hydrophobic lipids in the fur of aquatic mammals, such as this river otter, protect them from the elements. A fat molecule, such as a triglyceride, consists of two components, glycerol and fatty acids. Glycerol is an organic compound with 3 carbon atoms, 5 hydrogen atoms, and 3 hydroxyl groups. Fatty acids have a long chain of hydrocarbons to which an acidic carboxyl group is attached, hence the name fatty acid. The number of carbons in the fatty acid may range from 4 to 36. Most common are those containing 12 to 18 carbons. In a fat molecule, a fatty acid is attached to each of the 3 oxygen atoms in the hydroxyl group of the glycerol molecule with a covalent bond. Figure 2.18. Lipids include fats such as triglycerides, which are made up of fatty acids and glycerol, phospholipids, and steroids. During this covalent bond formation, 3 water molecules are released. The 3 fatty acids in the fat may be similar or dissimilar. These fats are also called triglycerides because they have 3 fatty acids. Some fatty acids have common names that specify their origin. For example, palmitic acid, a saturated fatty acid, is derived from the palm tree. Arachidic acid is derived from Arachis hypogea, the scientific name for peanuts. Fatty acids may be saturated or unsaturated. In a fatty acid chain, if there are only single bonds between neighboring carbons in the hydrocarbon chain, the fatty acid is saturated. Saturated fatty acids are saturated with hydrogen. In other words, the number of hydrogen atoms attached to the carbon skeleton is maximized. When the hydrocarbon chain contains a double bond, the fatty acid is an unsaturated fatty acid. Most unsaturated fats are liquid at room temperature and are called oils. If there is one double bond in the molecule, then it is known as a monounsaturated fat, example olive oil. And if there is more than one double bond, then it is known as a polyunsaturated fat, example canola oil. Saturated fats tend to get packed tightly and are stored at room temperature. Animal fats with stearic acid and palmitic acid contained in meat and the fat with butyric acid contained in butter are examples of saturated fats. Mammals store fats in specialized cells called adipocytes, where globules of fat occupy most of the cell. In plants, fat or oil is stored in seeds and is used as a source of energy during embryonic development. Unsaturated fats or oils are usually of plant origin and contain unsaturated fatty acids. The double bond causes a bend or a kink that prevents the fatty acids from packing tightly, keeping them liquid at room temperature. Olive oil, corn oil, canola oil, and cod liver oil are examples of unsaturated fats. Unsaturated fats help to improve blood cholesterol levels, whereas saturated fats contribute to plaque formation in the arteries, which increases the risk of a heart attack. In the food industry, Oils are artificially hydrogenated to make them semi-solid, leading to less spoilage and increased shelf life. Simply speaking, hydrogen gas is bubbled through oils to solidify them. During this hydrogenation process, double bonds of the cis conformation in the hydrocarbon chain may be converted to double bonds in the trans conformation. This forms a trans fat from a cis fat. The orientation of the double bonds affects the chemical properties of the fat. Figure 2.19. During the hydrogenation process, the orientation around the double bonds is changed, making a trans fat from a cis fat. This changes the chemical properties of the molecule. Margarine, some types of peanut butter, and shortening are examples of artificially hydrogenated trans fats. Recent studies have shown that an increase in trans fats in the human diet may lead to an increase in levels of low-density lipoprotein, LDL, or bad cholesterol, which in turn may lead to plaque deposition in the arteries, resulting in heart disease. Many fast food restaurants have recently eliminated the use of trans fats, and U.S. food labels are now required to list their trans fat content. Essential fatty acids are fatty acids that are required but not synthesized by the human body. Consequently, they must be supplemented through the diet. Omega-3 fatty acids fall into this category and are one of only two known essential fatty acids for humans, the other being omega-6 fatty acids. They are a type of polyunsaturated fat and are called omega-3 fatty acids because the third carbon from the end of the fatty acid participates in a double bond. Salmon, trout, and tuna are good sources of omega-3 fatty acids. Omega-3 fatty acids are important in brain function and normal growth and development. They may also prevent heart disease and reduce the risk of cancer. Like carbohydrates, fats have received a lot of bad publicity. It is true that eating an excess of fried foods and other fatty foods leads to weight gain. However, fats do have important functions. Fats serve as long-term energy storage. They also provide insulation for the body. Therefore, healthy, unsaturated fats in moderate amounts should be consumed on a regular basis. Phospholipids are the major constituent of the plasma membrane. Like fats, they are composed of fatty acid chains attached to a glycerol or similar backbone. Instead of 3 fatty acids attached, however, there are 2 fatty acids, and the third carbon of the glycerol backbone is bound to a phosphate group. The phosphate group is modified by the addition of an alcohol. A phospholipid has both hydrophobic and hydrophilic regions. The fatty acid chains are hydrophobic and exclude themselves from water, whereas the phosphate is hydrophilic and interacts with water. Cells are surrounded by a membrane, which is a bilayer of phospholipids. The fatty acids of phospholipids face inside away from water, whereas the phosphate group can face either the outside environment or the inside of the cell, which are both aqueous. Steroids and waxes. Unlike the phospholipids and fats discussed earlier, steroids have a ring structure. Although they do not resemble other lipids, they are grouped with them because they are also hydrophobic. All steroids have 4 linked carbon rings, and several of them, like cholesterol, have a short tail. Cholesterol is a steroid. Cholesterol is mainly synthesized in the liver and is the precursor of many steroid hormones such as testosterone and estradiol. It is also the precursor of vitamins E and K. Cholesterol is the precursor of bile salts, which help in the breakdown of fats and their subsequent absorption by cells. Although cholesterol is often spoken of in negative terms, it is necessary for the proper functioning of the body. It is a key component of the plasma membranes of animal cells. Waxes are made up of a hydrocarbon chain with an alcohol group and a fatty acid. Examples of animal waxes include beeswax and lanolin. Plants also have waxes, such as the coating on their leaves that helps prevent them from drying out. In our next section, we will go to Proteins.